Chapter 7 is all about the structure and function of the plasma membrane. So several things about the plasma membrane we've already talked about with the context of the structure and function of the cell and the structure and function of phospholipids. So the first part of this is really just a review. So there's a couple different ways we can describe the cell membrane. So one way to describe it is as a phospholipid bilayer. So remember, phospholipids have a polar head group with a phosphate and two hydrophobic tails. And so they self-assemble into membranes. The hydrophobic tails bunch together to get away from water, and the polar head groups face out towards water, eventually circling all the way around and making a double layer membrane, or a phospholipid bilayer. So another thing about lipids is they don't form polymers. So even though the membrane is made of these phospholipids, they're not permanently attached to one another. And that means that small molecules can actually squeeze in between the phospholipids. So another way to describe the cell membrane is that it's semi-permeable. It lets some things through, but it blocks other things from moving across the membrane. And the smaller and more hydrophobic something is, the easier it is to get through, whereas larger polar things are slowed down and charged ions can't get through at all. Okay. Another way to describe the cell membrane is a fluid mosaic. So you can think of each of the phospholipids as well as the proteins that are embedded in the membrane as individual tiles in a mosaic. But they're not cemented into place. They can move around past one another. They can slide laterally next to one another. And that's exactly what happens. If we look at how frequently phospholipids are moving in the membrane, it's really much more like a liquid than it is like a solid. They're constantly moving back and forth, 10 to the 7th times per second, sliding past one another. However, what doesn't happen very frequently is these polar head groups being able to flip all the way across the membrane into and out of the cell. So the outside of the plasma membrane and the inside of the plasma membrane really maintain their orientations and are distinct from one another. One piece of evidence we have for the fluid mosaic model for cell membranes comes from hybridoma studies. So scientists are able to take mouse cells and mark proteins on them with one color fluorescent marker, and take human cells and mark proteins on them with a different color fluorescent marker. And then you can actually cause these cells to fuse together by electrocuting them, actually, and it'll form one big cell, one hybridoma. Now, this hybridoma is not going to be able to replicate. You can't make like human-mouse hybrids this way, but we can do it to study the behavior of cells and cell membranes. And so initially when the cells are fused, the mouse proteins are on one side and the human proteins are on the other side. But after less than an hour or so, the proteins have scattered all over the place. They're not restricted from one side or the other side of the cell. How fluid and how permeable the membrane is influenced by what kinds of fatty acids are present in the phospholipid tails. If you have saturated fatty acids, those tails are pretty much straight and linear, and the phospholipids can pack close together and form a more solid, less permeable, more viscous membrane. And it's harder for small molecules to move across here. On the other hand, if you have unsaturated fatty acids in the phospholipid tails, those double bonds kink the tails sideways and push the phospholipids further apart from one another. That makes the membrane more fluid, less viscous, and more permeable. It's easier for small molecules to squeeze across the cell membrane. Another factor that influences membrane fluidity is cholesterol. But the effects of cholesterol vary depending on the temperature. So if you remember, cholesterol is a lipid made of 4 fused rings of hydrocarbons with a little hydroxyl group sticking out on the end. And it's a structural component of our cell membranes as well. The overall shape of a cholesterol molecule is kind of kinked. It's kind of bent and crooked like those unsaturated fatty acids. So at cool temperatures, the presence of cholesterol in the membrane actually keeps the membrane fluid and permeable. On the other hand, because cholesterol is made of these 4 fused rings, the molecule itself is not very flexible. And so at warmer temperatures, cholesterol actually makes the membrane more solid or more viscous. And we see this in cholesterol rafts on cell membranes that have a high concentration of cholesterol where proteins are anchored. Cell membranes are made mostly of phospholipids. By number, they're the most abundant molecules in our cell membrane. But there are also proteins that are part of our cell membranes. There are some proteins that are associated just on the surface of the plasma membrane, so they attach just to the phospholipid head groups. And those are called peripheral proteins. But proteins that go through or across the membrane are called integral membrane proteins. They integrate, they go into the hydrophobic core. Okay? Thinking back to the last unit, these proteins that are embedded in the membrane are made by ribosomes where? They're made by ribosomes that are on the surface of the rough ER. And those ribosomes push those proteins through the membrane. The membrane proteins that are integral are amphipathic. They have hydrophobic parts that interact with the hydrophobic tails of the membrane, and they have hydrophilic parts that stick out either to the outside of the cell or the inside of the cell. Any protein that sticks into the hydrophobic part is called an integral membrane protein, but if it goes all the way across, if it has parts of the protein exposed on the extracellular surface as well as the inside of the cell, those are called transmembrane proteins. These membrane proteins have an incredible number of functions. Just a couple of examples that we've already talked about. We talked about proteins that anchor cells to each other and to the extracellular matrix. There are proteins that catalyze enzymatic reactions at the surface of the cell. There are proteins that receive signals from the outside. There are proteins that are transport proteins, and those are the ones we're going to talk about the most, as well as ones that are involved in cell-to-cell recognition. Reminder, all of our transmembrane proteins, where they started from was ribosomes on the surface of the rough ER that pushed those proteins across that membrane. Then those vesicles pinched off from the rough ER, those glycoproteins got modified in the Golgi and then transported to their final destination. So I think this picture does a really good job of reminding you that the blue, the inside of the rough ER, is the inside of the Golgi, is the inside of the vesicle, which ends up being the outside of our cell. On the other hand, anything that happens in the cytoplasm, any proteins made by cytosolic ribosomes, remain in the cytoplasm. So our membrane is semi-permeable. It's also selectively permeable. It lets some things through and blocks other things from coming across. So the smaller and more hydrophobic something is, the easier it is to get across the membrane. The larger and more polar something is, the slower it is to cross the membrane. But there are all kinds of charged ions that our cell needs that have to come into and out of the cell, and those have to be transported. So we're going to talk about the different kinds of transport across the membrane. We'll talk about direct transport by diffusion as well as active transport that requires a source of energy. So diffusion is the movement of molecules down their concentration gradient. So if we just imagine we have a beaker of water that has a semi-permeable membrane and we put dye on one side of the membrane. These molecules are going to bounce around, and some of them are going to stay on this side, but some are going to make their way across and bounce to the other side. And over time, the molecules are going to move back and forth until they spread out equally on either side of the membrane. At that point, when the concentration of molecules are the same on both sides of the membrane, we've reached equilibrium. Now, that doesn't mean the molecules stop moving. They're going to continue to move, but they're not moving in any one direction versus the other. Whereas at the beginning, they were moving from the left to the right. If we had different types of molecules, each one would diffuse down its own concentration gradient. So the yellow balls are going to diffuse to the right, and the purple balls are going to diffuse to the left. Just a review of a couple of terms that we introduced in Chapter 3 on water. The solute is the thing that's dissolved, so our dye particles or our salt ions. The solvent is the dissolving agent. So in biological systems, that's usually water, and then the mixture of the two is the solution. And when we talk about different solutions, we can describe the relative solute concentration between two. So a solution that's hypertonic has a higher solute concentration compared to other solutions. So these terms are relative. So the word hypertonic describes something compared to another solution. Just like you could say that chair is bigger than a different chair. Okay? You couldn't just say it's bigger. Well, it's bigger than what? Okay? That's the same with hypertonic. Hypertonic means it has more solute than some other solution. Okay? Hypotonic, on the other hand, means it has a lower solute concentration, a lower salt concentration compared to some other solution. And isotonic means that they have the same concentrations. So molecules are always going to diffuse down their own concentration gradient from an area of high concentration to an area of low concentration. That's true of our salt ions if the membrane is permeable to them, but it's also true of water. Water will also diffuse down its own concentration gradient, and the diffusion of water is called osmosis. Because cell membranes are permeable to water and not permeable to salt ions, this is going to influence what thing diffuses across the membrane. And we can actually use this principle to gently lyse cells open in the lab by using the diffusion of water. So in this image, we have a U-shaped tube that has a low solute concentration on this side and a high solute concentration on this side. And I've kind of drawn the same thing here on the board. So this has a high solute concentration. This has a low solute concentration. Now, if we assume that the membrane were permeable to the solute, if the salt could move, the salt would move from high solute concentration to low solute concentration. That would be diffusion of the solute. But in this case, this membrane is not permeable to the solute. It's only permeable to water. If we think about what that means, on this side, if we take out this volume, everywhere there's a black spot, we can imagine there's no water. So this actually has a low water concentration per volume, and this has a high water concentration per volume, right? Here, it's almost all water, there's just one little black spot. Okay? So water is going to diffuse down water's concentration gradient, and water is going to move from the left to the right. Diffusion of water is called osmosis. Okay? If we use our vocab words to describe this, this side is called the hypotonic solution. It has a low solute concentration, which is the same thing as saying it has a high water concentration. This side is the hypertonic solution. It has a high solute concentration, which is the same thing as saying it has a low water concentration. And what happens in this scenario is osmotic pressure, the diffusion of water by osmosis, will actually cause the water to move to the right, and it will appear to defy gravity, and the water will actually raise up on this side of the tube. This is the same principle that's used for dialysis. There's only 3 possible questions that I could ask you guys about this type of situation. What happens when you put a cell in a solution that has a lower solute concentration, an equal solute concentration, or a higher solute concentration? So in this first case, the environment is hypotonic, which makes the cell hypertonic compared to its environment. Okay? If the membrane were permeable to the salt would move out of the cell. But membranes are not typically permeable to salt ions, so instead the water is going to move by osmosis into this cell. In this case, the cell and the environment have the same concentration, so they're both isotonic to each other, and water is going to move both into and out of the cell, but in neither net direction. In the last case, the environment is hypertonic compared to the cell, and the inside of the cell is hypotonic compared to its environment. If the salt could move, the salt would move into the cell, but since cell membranes are not typically permeable to salt, the water is going to move out of the cell. And the consequence of this is going to be different whether it's a plant or animal cell. Plant cells have a cell wall which limits their uptake of water. The direction of water's movement is going to be the same whether it's in a hypo or hyper or isotonic solution. But plant cells will just fill up with water and become turgid if they're in a hypotonic solution. Animal cells will burst. Animal cells are happy in an isotonic solution, plant cells are limp, and both kinds of cells will shrivel up when they're in a hypertonic environment. This is particularly bad for plant cells because their plasma membrane gets ripped away from the cell wall and they become plasmolysed. This is actually one of the trickier concepts in Unit 2, so I highly encourage you to do the homework on Mastering Biology to practice these concepts. I guarantee there will be questions like this on the exam.